Okay, so are there any questions before we start today? So notice that the final exam is on Monday at 8 o'clock in the morning. That's the schedule. So we'll have some time next week if you have particular questions about the review sheet. So I feel like you should make a point of going over the review sheet and then going over your notes in your textbook. And then if you have any questions, you can let me know. Okay, so we were talking about early embryonic development. And remember that fertilization has to take place in the oviduct or the uterine tube. And that sometimes that early embryo can split into two to form a twin. So if we look at the two different types of twins we can have a twin that is said to be dizygotic. So what this means is that there's two different zygotes. So this means that two oocytes were ovulated. And each was fertilized. So just think di means two, okay? So this is different from monozygotic. So in monozygotic, we have one oocyte is fertilized, and then the embryo splits during early embryonic development. And that embryo forms two separate individuals. Okay, so mono means one versus di, which means two. So which one of these would you say it would be an example of identical twins? Monozygotic. So this is identical twins. So these would be called what? They're not identical, they're called fraternal twins. Now, there is also the possibility of embryos, when we talk about the dizygotic twin, there's a possibility that embryos could fuse. And this is what is referred to as a chimera. So a chimera is where more than one embryo is produced. And this actually is thought to occur more frequently than was originally thought. So they think that one out of 8 pregnancies, you actually start out with more than one embryo. And so that's a lot, right? And so generally what happens is that one of the embryo dies and is reabsorbed, but sometimes the embryo can fuse to form an individual that has more than one type of cell type. So more than one genetic cell type. So the embryos fuse and produce one individual. With two different cell lines. Right? So some of their body could be one cell line, and then the other part of their body could be another cell line. And so this came up recently in the news because of paternity testing. So there was an example of a male who failed a paternity test. So when they went in and they tried to determine whether the offspring was his, and the reason why he actually took the paternity test is because they had a fertility clinic that was doing in vitro fertilization. So they were actually, the egg and the sperm were actually fertilized outside of the female's body, and then the embryo was put back in. And then the baby came out a blood type that was not predicted. So the baby was not logically could possibly be from those two parents. It's a little bit more complicated than that. But then when they did the paternity test, they found that what they thought the father was, he was actually a brother. And so what this suggests is that the sperm's DNA was of one embryo, and then his DNA taken from his saliva was of another embryo. And so we don't know exactly where, what cells are of what individual and what cells are another individual, but there's this idea that this could be more common than previously known. And there's actually a person that I know in the community that is a chimera, and she has some health issues because of it, because the immune system sometimes doesn't or detects those cells as being foreign. So there's some autoimmune issues with her.
I saw that, but with the eyes. One blue eye and one brown eye.
So that could be a possible example of a chimera. It could also have something to do with development and the way that the melanin is deposited in the iris. So it's not necessarily chimera. Yeah. But sometimes females could have, like, for example, a female could actually have a male genotype in some of her cells if the twin that she took in was male. So you could actually have an individual that had some female and some male cells. Yes?
This might sound kind of dumb, but I have read somewhere that this girl was giving birth to a baby, but the baby had both one guy's and another guy's DNA thing.
It could be that what happens there is that she gave birth to two babies?
No, just one.
Okay, just one.
He had both of them.
I don't know how that would be possible. I'll have to think about that.
They had put it on there.
I guess it could be possible if that baby that was born was a chimera.
Oh.
So let's say she ovulated 2 oocytes, and then she had sperm from 2 different males in her reproductive tract, and the 2 oocytes got fertilized, right? And then the embryos fused, and then the baby was born a chimera.
That makes sense.
So essentially a chimera is just an embryo, or it's just a baby that could eat the other baby?
Well, sometimes they call it the vanishing twin syndrome, right? So sometimes what happens is that early on during pregnancy, they detect that there's twins, and then for some reason that twin vanishes. And it could be that the twin just died and was reabsorbed, by the mother, but it could be that the twin that they actually fused, okay? So you have a situation that's sometimes called the vanishing twin. Right, and so this is where one twin disappears during pregnancy. And it could be a chimera, but not necessarily. Okay. I also had a student who said she was born and she actually had a growth on the side of her head. And when they biopsied it and removed it, it was actually her twin. Right, yeah. Okay. Okay. So let's move on to normal or your typical embryonic development. And we'll talk about implantation. So implantation starts at 7 days. So that would be 1 week. And what implants is called the blastocyst. And so remember that the blastocyst at this point is free of the fertilization membrane. And it consists of a ring of cells called the trophoblast. So that would be a ring of cells. And then we have cells inside, which is called the inner cell mass, right? So these would be cells along the outside. So tropho means food. And so this trophoblast is actually going to become part of the placenta. So it's going to help to feed the, it's going to help to feed the developing embryo. So tropho means food. And then this inner cell mass is what is going to become the embryo. So the way that this works is that the trophoblast adheres to the endometrium, and then it releases enzymes, and it digests its way into the lining of the endometrium. And then we have this inflammatory response that happens. So the mother's immune system detects that something is invading the tissue and destroying it. And so the inflammation causes blood and white blood cells and all kinds of fluid to go there. And then what eventually happens is you get a cyst forming. So the endometrium actually forms over the trophoblast, and then you have the blastocyst implanted in the lining of the endometrium. So this would be the blastocyst digests its way into the endometrium of the uterus. And then it is covered over by the endometrium. Okay. So if we look at how this looks from a cellular point of view, okay, so this is my embryo. So this would actually be a picture of one. This is my inner cell mass, right? And then what happens is that it invades the endometrium, so it starts to digest it and actually destroys some of the cells, and then it eventually becomes incorporated. Excuse me. Eventually becomes incorporated. Oops. Do I not have that picture? Oh, I don't have that picture. So it'll eventually become covered over and be incorporated into the lining of the uterus. So remember that this early embryo has to secrete HCG in order to be maintained. So HCG actually maintains the corpus luteum in the ovary and prevents prevents the endometrium from sloughing off. Okay, so this shows when HCG is most produced. And so you'll notice that it starts being produced, and then at about 12 weeks, it drops off. And 12 weeks is when we get the development of the placenta. And so the placenta takes over. So at 12 weeks, the placenta starts to produce estrogen and progesterone, and that maintains the lining. Okay, so early on, the embryo produces HCG. Oops. Right. And this prevents the corpus luteum from degenerating. And it is the corpus luteum that produces estrogen and progesterone. Right. And remember, the corpus luteum is actually in the ovary. Okay. At 12 weeks, we have the placenta. So the placenta takes over and produces estrogen and progesterone. So the placenta is actually an endocrine gland also, besides being a way that the embryo gets nutrients from the mother. So it produces its own hormones. Okay, so if we look at the extraembryonic membranes that are formed during the development, we can look at the inner cell mass and the anatomy of it. The inner cell mass has an epiblast to it. So epi means on top. So the epiblast actually becomes the amnion. And the amnion is the membrane that holds in the water, because embryonic development has to take place in a water environment, right? So the amnion is that. So this is the amniotic sac filled with amniotic fluid. So embryonic development has to take place in an aquatic environment, right? So we create one inside the mother. So the amnion is one of those extra embryonic membranes. It actually does not become part of the embryo, right? It's left behind when the baby is born. And then we also have a hypoblast. And the hypoblast is below, and it results in the yolk sac. And so the yolk sac contains nutrients, but in us, unlike in chickens and other animals that have eggs, it's not our sole form of nutrition. And so it actually serves other functions. So it actually becomes part of the primitive gut. In the embryo. So the gut, the digestive tract, okay, so the yolk sac, okay? And it also forms, let me see, make sure I get that right. I thought it formed, oh, it's the source of earliest blood cells and blood vessels. And so if you remember from our video that we watched, blood is one of the circulatory system is one of the first things that arises because of the need to get nutrients and oxygen to all of the cells in the multicellular embryo. So that is a really early system that develops. And so, even the heart, develops early on. And so sometimes they'll go in and they'll check to make sure that even in the earliest embryo, like 8 weeks, they can see a heartbeat, right? And if they don't see a heartbeat, then they know that it is an inviable pregnancy. And so sometimes they'll have to go in and they'll have to remove it through a DNC or a dilation and curvature, right? So they go in and they actually remove the embryo if it is not spontaneously aborted by the mother. Okay. So if we look at the structures here, yellow is always the yolk sac. The blue is always the amnion, right? So epi means on top, hypo means below, right? And then right in the center between these two is the kind of the embryonic disc. And that embryonic disc is going to form all of the tissues, the other tissues in the developing embryo. Okay. Now there's one more extra embryonic tissue, and that is called the allantois. So write that one down. So we have the amnion, we have the yolk sac, and then we have the allantois. Actually, there's a fourth one, but the allantois forms the umbilical cord. Okay, and then we have the chorion. And this forms part of the placenta. So this forms the embryonic part of the placenta. This is the diagram that shows how we have inflammation, right? The blood vessels, there's actually a pool of blood here, and then the endometrium just fuses over the outside of the embryo. Okay, so when we look at the development of the placenta, this is called placentation, right? And so this develops and is fully functional at 12 weeks, right? So functional at 12 weeks about. Right? And it has two parts. So the maternal part of the placenta portion is called the decidua basalis. Right? So when you look at the placenta, you can see the difference between the, if you've ever seen one being delivered, you can see the difference between the fetal side and the maternal side. The decidua basalis is really smooth, right? And it actually is called decidua because it sloughs off, right? So when the baby is born, the placenta also has to be delivered. So after the baby is born, then you have continued contractions, uterine contractions, that then cause the placenta to move off the endometrium and come out through the birth canal. So decidua just means that it will fall off, right? Decidua basalis, okay? And then when we look at the fetal part of the placenta, we have what is called the chorionic villi. Okay. Now there's another part that's actually not part of the placenta, but is actually important because it's maternal tissue that encapsulates the developing fetus. And this is called the decidua. So this is, I'll just put this in parentheses. The decidua capsularis is maternal tissue that envelops the embryo. So it's actually not part of the placenta in terms of like transfer of nutrients and oxygen, but it's part of the structure of the developing fetus inside the mother. Okay, so if we look at this, okay, so this is the decidua basalis right here, right? So that would be maternal. So these are actually maternal blood vessels coming in. And then we have the chorionic villi, which are capillaries that extend into these like spaces. The maternal blood vessels, and we saw this in the video, the maternal blood vessels put blood into these spaces. And then we have the diffusion of nutrients and antibodies and other possible bad stuff crossing over and coming into the fetus, right? So you can see all the structures here. So this is the capsularis is around it. The decidua capsularis is around the embryo. The blue would be the amniotic membrane. This would be the yolk sac, right? And then we can see, I don't think they have the allantois on here, but the allantois actually becomes part of the umbilical vein and artery that delivers blood to the fetal part of the placenta. So when we look at the placenta, we see that there is a barrier, right? So the placenta is what transfers nutrients, oxygen, and antibodies to the fetus. But there is a barrier, right? So that typically things it's protected, right? So things don't tend to move through the placenta from the mother to the fetus. But we now know that this barrier sometimes lets stuff through, right? So this is a barrier that can prevent some things in mother from reaching the fetus. We did talk about a blood type, Rh, right, a factor, an Rh antibody. So if the mother is producing Rh antibodies, they can get through the placenta, right? And they can actually cause hemolytic anemia. So what that means is that the antibodies get through the placenta and go into the embryo, and then they bind to fetal red blood cells and cause them to be broken down, right? So we talked about that when we were talking about blood typing. So the antibodies can make it through. The other thing that can make it through would be alcohol. So we have alcohol able to get through this, and alcohol is considered a teratogen. So a teratogen is a chemical that can cross the placenta barrier and cause birth defects in the developing fetus. Okay, so alcohol would be one of these. We also know now that the Zika virus, I don't know how to spell that. Is it just Z? I think it's just Z. Zika virus, right, causes microcephaly. So that would be an example of a teratogen. But the most famous example of a teratogen was a chemical called thalidomide. Okay, and so in the 50s and the early 1960s, they were giving females thalidomide during early pregnancy, like for morning sickness. So it was kind of a sedative, and it was a treatment for morning sickness, but it was a drug. And it caused dramatic birth defects, and specifically, it caused the development of deformed appendages, right? And so in the 1950s and early 60s, there was about 10,000 babies affected by thalidomide. And so they were born with things like hands attached to their shoulders, right? Or deformed feet, so their feet attached directly to their torso. And so it had something to do, it was affecting the development of their appendages. And so that is sometimes referred to as the thalidomide generation. And it was just a real horrible thing that happened, kind of similar to what is happening with the Zika virus. These people are normal mentally though, so they have the same mental capacity as a normal individual, just deformed appendages. So this was a big deal, and they kind of banned thalidomide from being used ever again until recently when they wanted to start using it for cancer patients. And so it seems to be a good drug for cancer patients. And so there was a big uproar about people, whether or not, since it caused just this horrible thing to happen, whether it should even be used medicinally again. And I'm not sure what happened with that, if they said that it was okay to use it for cancer. Okay. So those are teratogens, and that's early embryonic development. Okay. This is another diagram that shows that chamber where we have the exchange of oxygen and nutrients, right? We did talk about how sometimes things can go from the fetus into the mother. So we now have evidence that fetal cells can sometimes slip across this capillary and get into maternal circulation. And remember, we listened to that video about whether or not those fetal cells were good, because it's like being injected with fetal stem cells, or whether they were causing autoimmune responses in the mother. Okay, so we can start talking about how cells differentiate. And so the important idea here is that we are all, at one point in time, a single cell. So all cells in the body have the same genetic material, except for if you're a chimera, right? In the body have the same genetic material. Right? And if a cell can become any type of cell, it is undifferentiated, and it is generally referred to as a stem cell. So a stem cell is unspecialized. It's undifferentiated. So it can actually become anything in the body. Okay. And then as the stem cells start to mature, so early embryonic development, everything's a stem cell. It can become anything. But at some point, the cells start to differentiate, and what happens is that genes are turned on and off, and the cells become from unspecialized to specialized. So gene regulation, which can be described as the turning on and off of genes, causes cells to specialize. And sometimes they specialize so much that they, can't even reproduce after that. So if you think about red blood cells, right? So we have stem cells in our bone marrow that give rise to the red blood cells, but they specialize so much they even eject their nuclei, get filled with hemoglobin, and then they're just specialized, right? And then they die, right? So that has to do with gene regulation. So it's interesting to note that all of our cells in our body could produce hemoglobin because all the cells in our body have the gene that codes for hemoglobin, but only the red blood cells do, right? So only the red blood cells have that gene turned on, okay? So when we look at this early cell differentiation, we can talk about germ layers. So this is the first instance where we start to get specialization. And these are actually called embryonic. I'll put embryonic germ layers. Okay. So there's 3 layers. We have the ectoderm, which is the outer layers. Think ecto means outside, right? We have the mesoderm, which is the inner layer. And then we have the endoderm, which is the innermost layer. So mesoderm is middle. So think meso, think middle. Okay. Endo is inside. So these are in the embryo, and what it means is that the cells start to become specific tissues. And so when we look at the ectoderm becomes the skin, right? It also becomes the nervous system. So our brain and our spinal cord are ectoderm in origin. I'll explain that in a minute. And I'm going to skip over mesoderm. The endoderm is like the epithelial tissue that lines our organs. So this lines the organs, like the digestive system, the respiratory system, and it also forms the glands. Okay. So mesoderm becomes muscle, bone, for example, and everything else besides what is produced by the ectoderm and the endoderm. So our vertebrae, for example, are mesoderm in origin, whereas our spinal cord is ectoderm. Now in your book, these all have a color. So ecto is always blue, meso is always red, and endo is always yellow. So if we look at a diagram that shows this early embryonic development. So in the middle of the early embryo, where we have the epiblast and the hypoblast, we have the embryonic disc, right? And here it is developed into ectoderm and endoderm. And then we get some cells migrating into the middle, and that becomes the mesoderm, right? And so if we look at how this progresses, oops, wrong way. Okay. If we look at how this progresses, we can see that we get the development of the nervous system from the ectoderm. So what happens with the ectoderm is it actually invaginates and it produces a neural tube. So sometimes you hear about neural tube deformities in babies. So an example of a neural tube defect is what? What's a really common example of a neural tube? Yeah, what is it? Yeah, so spina bifida would be an example of a neural tube defect, right? So that means that the neural tube did not form properly, okay? And then notice how the mesoderm wraps around that neural tube. And so that's going to be the vertebrae. And so the vertebrae wrap around and they protect the neural tube. And then notice how we have the lining, we have muscles, but we also have the digestive tract. So this is my early digestive tract, and so this is endoderm. So what happens is that these cells, wherever they find themselves in the embryo, that turns on genes and turns off other genes, and then the cells start to differentiate, okay? And so this differentiation can lead to the development of organs. So this is just a diagram showing all the endoderm in an embryo, So you can see even at 5 weeks, we have the development of the liver and the pancreas, which are glands, right? The gallbladder is also is part of this, right? You have the development of the right and the left lungs, right? The development of the pharynx, the respiratory, upper respiratory tract, okay? So that would be cell differentiation. And there is a book, or excuse me, there's a diagram in your book that shows, right, everything that is derived from ectoderm, everything that is derived from endoderm, and then mesoderm. So there is a essay question that is related to this, okay, this idea. Okay, so if we look at the fetal circulation, so the umbilical cord is fetal in origin. And if we looked at a cross-section of the umbilical cord, it kind of looks like this, where you have 2 umbilical arteries, and then you have a big umbilical vein. So it looks like a face, okay? So this is my vein, my umbilical vein. And this is my umbilical arteries. Okay. This is contained within the umbilical cord, and this actually has a name. All this stuff that's surrounding the arteries is connective tissue, and it actually has a name. It's called Wharton's jelly. Right. So this is connective tissue, and this connective tissue has stem cells in it. And so one thing that you can do if you have money, right, is you could actually, because it costs money, you could actually take your baby's umbilical cord and pay a company to store the stem cells from it, so that if your baby ever needed stem cell therapy, they would have stem cells available, right? You can also donate your umbilical cord for other people to harvest the stem cells from it to be used in stem cell therapies, okay? Okay, so these are fetal in origin. The arteries, however, are carrying unoxygenated blood. So this is that weird example, because arteries are carrying blood away from the fetal heart. So by definition, it is an artery, but it is carrying unoxygenated blood, okay? So the umbilical vein is carrying oxygenated blood, because it's carrying the blood that has already gone to the placenta and then is coming back to the fetus, and it's going to come back to the fetal heart. Okay. Now, in the fetal circulation, we're not getting any oxygen from the lungs. So fetal blood must bypass the fetal lungs. Right? So we don't want blood going into the lungs because there's no oxygen there, right? And so does anybody remember what structure in the heart causes blood to go from the right atrium to the left atrium without going into the pulmonary circuit? Yes, so the foramen ovale, right? So the foramen ovale. Okay. So this shunts blood from the right atrium to the left atrium, and you bypass the pulmonary circuit. Now, when a baby is born and it takes its first breath, what we want to have happen is that we want that valve to, there's a flap, and we want it to go and shut that off, right? Because when a baby takes its first breath, the blood then has to be shunted to the lungs, right? So upon birth, the blood must be shunted to the lungs. right? So we hope that the flap covers over the foramen ovale, right? And if we just make it closed, so we close the foramen ovale. Okay, so it's a little bit more complicated than that, but that's what you need to know for the final. Oh, do I not have that picture? Oh, here it is. Okay, so this shows the umbilical artery and the umbilical vein. So what's that big one? What's the big red one? Is it a vein or an artery? Vein. Vein. It's a red vein. Okay, one of them. Notice how there's two umbilical arteries, right? Okay, so the umbilical vein comes back and it goes to the liver. So when we were doing that, the dissection of the fetal pig, remember that that goes to the liver, right? And then it then goes into the circulatory system and it goes to the heart. But we want the blood to go right from the right atrium, right, into the left atrium and then to be pumped out to the body, okay? So this is the kind of the difference in the circulatory system between the fetus and the newborn. So upon birth, these have to constrict, right? So you have to stop blood flow in the umbilical artery and the umbilical vein. Okay, so those constrict and they actually form ligaments. So those leftover blood vessels turn into connective tissue. Okay. Okay. So let's talk about the effects of pregnancy on the mother. Okay, so when we think about pregnancy, we can talk about the physical effects, but we can also talk about the physiological effects. One thing that happens is that the uterus enlarges and it puts pressure on the diaphragm. Also on the stomach and the bladder, right? So when a female is very pregnant and the uterus is very big, sometimes it's hard for them to breathe, right? Because they feel like they're out of breath all the time because the uterus is actually pushing up on the diaphragm. And then it pushes on the stomach, and sometimes you get acid indigestion, right? So you get the stomach and the esophagus and that diaphragm, that all that, you know, acid comes up into the esophagus. So you get gastroesophageal reflux. And then bladder means that you have to urinate all the time. Then the female, they are producing more urine because of pregnancy, but just the pressure of the developing embryo on the bladder also causes problems, right? So this is the enlargement, right, of the uterus as the baby develops, okay? Okay. We also have an increase in the amount of blood. And so by 37 weeks, it's something like, I think it's like something like 40% more blood. Let's see if I can find my notes here. %40 more blood in the female circulatory system. Oh, where is it? Oh, I can't find it. Okay. So we'll say at 37 weeks, about 37 weeks of pregnancy, I'll just wing it, right? We have about 40% more blood. Okay. So what that means is that we also have an increase in kidney function, right? Because the kidney has to filter all this blood. So the kidney, increase in kidney function as it filters the blood, right? And also there is a problem possibly with an increase in blood pressure because of this extra blood. So we have a problem that is known as preeclampsia. And preeclampsia in a pregnant female is an increase in blood pressure. And this can become a big problem, right? And so when you have preeclampsia, one of the things they typically do in late stages of pregnancy is they make you stay in bed, right? And they make you regulate your blood pressure, and you're bedridden. And oftentimes they'll induce birth prematurely if blood pressure goes excessively high. So preeclampsia can lead to eclampsia, which can cause problems in the mother as well as the developing embryo. Okay, so if we look at the other things that happens in the pregnant female is that we get a problem, or possible problem, with gestational diabetes. So gestational diabetes is not found in everybody, right? But it is caused by an increase in glucose levels in maternal circulation. And I believe we talked about this when we were talking about other types of diabetes. Is that true? Okay. So this is actually caused by the embryo, and the embryo produces a hormone called human placental lactogen. And this binds maternal insulin. So this happens in everybody, right? But if the mother can't produce enough insulin, so if mother can't produce enough of the insulin, then it becomes gestational diabetes. And it's a really interesting type of diabetes because as soon as the mother gives birth and the baby's gone and it's not producing any more human placental lactogen, then the blood glucose levels go back down to normal. Okay. Okay. So let's look at birth. So the process of giving birth is actually called parturition. Parturition, right? So this would be labor. And I want to talk about how hormonally labor is initiated, right? So the initiation of labor actually occurs with the fetus. So the fetus causes the placenta, because there's a fetal portion of the placenta. So the fetus causes placenta to release high levels of estrogen. Okay. So these high levels of estrogen cause oxytocin receptors to be added to the myometrium of the uterus. So this causes, so before those high levels of estrogen, these receptors are not there. So the myometrium is the smooth muscle. So myo means muscle. So the myometrium of the uterus. Okay. So what happens then when oxytocin is released? So the fetus produces oxytocin. It binds to the myometrium, and it causes uterine contractions. Okay. These first uterine contractions are sometimes called Braxton Hicks. So this is false labor, is when the contractions are not synchronized, and they're called Braxton Hicks. Right. And so what it feels like is it just feels like a tightening. So when you're pregnant and your uterus starts to tighten up and put pressure on the fetus, but then it relaxes and they're not like, you know, coming one after the other and they're not getting stronger, then that is what is referred to as false labor. Okay. Now this whole process of going from false labor into true labor, so true labor would be when the contractions are synchronized and they build. Okay. So you can time them, right? How close together they are. So they're synchronized, they're close together. We'll put close together. And they get stronger. Okay, so that would be true labor. This process of true labor is an example, one of the few examples of a positive feedback mechanism. And so in this particular instance, the contractions would be the stimulus. So the contractions stimulate more oxytocin to be released, and this time it can be from the mother. So more oxytocin released, right? More oxytocin stimulates, contractions stimulate more oxytocin to be released. That'd be better, to be released, okay? And then the contractions, and then the increase in oxytocin causes stronger contractions. So in this case, it's positive because the stimulus and the response actually increases the stimulus, right? So it's not a negative feedback because we don't want homeostasis to be maintained, right? We want the contractions to build and get stronger until the baby is forced out of the uterus, okay? So in your book, there's a really nice diagram that shows this mechanism, right? So estrogen from the placenta, right, induces the oxytocin receptors to be put, right? And then oxytocin from the fetus and the mother then stimulates the placenta and the uterus to contract. It also, I didn't mention this part, but prostaglandins can also cause more vigorous contractions to be produced, right? And then this is a positive feedback because as the uterus contracts, it actually causes more oxytocin to be released. And so what this is going to lead to is the different stages of labor. The first being dilation. So you notice that the baby is in position here where they're pressing on the cervix. And so when we look at this first stages, we get dilation of parturition, right? And so dilation refers to the thinning and widening of the cervix. Must be next door. Okay. Okay. So when they say you're 2 centimeters versus 8 centimeters dilated, what they're looking at is the size of the opening in the cervix, right? So the cervix during pregnancy is a barrier, right? It has a cervical plug, right? And it prevents stuff from getting from the vagina up into the uterus. But in order for the baby to come out, that barrier has to be opened, right? So the baby can move out, okay? So if we look at how this occurs, remember that the big problem with our giving birth, humans in general, is that we have babies with huge heads. And they have to fit through the opening of the pelvis. And so it's the pelvic outlet. If you remember, the female's pelvis is shaped differently than the male's, and it's wider and broader. But in order for the baby to move through the birth canal, it actually has to rotate. So notice how it here is on its side, but it rotates, tends to rotate face down, right? So as it rotates face down, it is better able to fit through that. And then once the head crowns and the head comes out, it's pretty easy to give birth after that because everything else is smaller by comparison, right? So the shoulders are kind of all like this, right? But they come out. So one also big problem is this: is sometimes you get tearing of the muscle that surrounds the vagina, which here they show this is the perineum, right? And so sometimes they have to do an episiotomy. And so episiotomies, I don't know if they're popular now. When I was giving or having children, there was a question about whether it was better to actually cut the perineum, whether it would heal better, or whether or not you should like massage it and try to get it to kind of relax. And then if it ripped a little bit, then that was easier for it to heal than a big cut. Right, so does anybody work with pregnant females?
Yeah, her first baby, she got torn, and the second she had a episiotomy. Okay. She said the second healed a lot better.
Okay, so there's a kind of an argument whether or not we should be just letting it tear naturally, or whether or not we should cut. Okay.
The episiotomy said they don't.
They don't usually do it.
They only did it because the baby was losing, or its heart rate was decreasing because it was losing oxygen.
Okay, so I think what they try to do now is they try to massage it and get that perineum to relax so that if it does tear, it's not going to be like horrible tearing, right? And so then notice even once the baby is out and they cut the umbilical cord, we still have to get rid of the placenta, right? So this decidua basalis has to come off, right? And then the placenta has to come out. Okay, so that is parturition. Okay, so I think I'm going to stop there for today. And we're going to start next week on human genetics. So we're going to talk about molecular, we're going to talk about the human genome, and then we're just going to talk a little bit about patterns of inheritance. Okay, I can put it back. Contractions stimulate more oxytocin to be released.
I'll just cause it stronger.
And this increases in oxytocin, yeah.
Thank you. I think, is that the last, on the last question on our essay thing, are you wanting the positive feedback mechanism added into it too?